Thank you very much for accepting this invitation. We are very happy to have you here and welcome you to the 2023 TIC and MITES community webinar. My name is Gloria Isabel Giraldo Calderon, and I am from the University of Notre Dame. And today I am joined by other members of the ViewPath team. Sam Run from the University of Notre Dame, Sarah Kelly from the Imperial College of London, and David Bruce from the University of Pennsylvania, who is one of ViewPath project PIs. What are we going to talk about today? In the first part, I'm going to tell you what is ViewPath and Vectorbase, how you can contact us, get some help, documentation, and news, and give you a demonstration about Vectorbase. But the second part is the one that we are more excited about because we are very happy to have you here. We want your feedback. We want to hear from you what Vectorbase has to offer about TICs and MITES. What are we missing? How do you think we can improve the site? So it's really the part that we are looking to. This is the second time we have a specific TIC webinar. Last year we have another one, and I invite you to watch the recording from last year. We tried to make this different from the one from last year, and the other one also has very important information, but you know we don't want to take much time, so that's why we invite you to look at the one from last year because it also has topics that I think are very important for the TIC community. Okay, a little bit about the Bioinformatics Resource Centers for Infection Diseases. This is a program from NIH that started in the year 2004, and this is an open access resource and is online. We are currently 2 BRCs. One, the PPBRC, that, David, are you able to see my mouse in the screen? Okay, thank you. That focus on bacteria and viruses. And the other one is ViewPath, the one that we represent, that has eukaryotic pathogens, including fungi and parasites, arthropod vectors, and other invertebrates and hosts. And if you want to learn more about the BRCs, here is the link. Okay, now more into ViewPath. This is the homepage of our site. And if you look at the header, you will see all these icons. And these icons are actually links to the different sites that we have. The difference between them is the organisms they host. But the advantage is that they all have exactly the same layout, tools, and functionality. So if you learn how to use Vectorbase because you're interested in TICs and MITES, you already know how to use Paraplasma database where you will find Babesia, or you already know how to use host, where we have the genomes, for example, a study from guinea pigs in which they look at how a tick vaccine might work in a host. And if you look at the header, here it says organisms, and we have more than 700 genomes. So combined, all these sites up, we have more than 700 genomes. Today, we are focusing on one of them, which is Vectorbase. Vectorbase is the home of vectors of human parasites, viruses, and bacteria. And why I have underlined this is because in addition to vectors, we also have other arthropods that we have included for comparative purposes. For example, for mosquitoes, we have the vectors of malaria, Anopheles, but we also have another Anopheles that are not vectors. We also have the fruit fly, Drosophila melanogaster, because we know it's very useful for comparisons. We also have this snail here, which is an intermediate host for Schistosoma. So we have, in addition to vectors, we have other species of interest. What type of data we have? 2, omics and field collected population data, and I'm going to give you more details about that. So here we have kissing bugs, we have stable flies, housefly, other mosquitoes, sunflies, tsetse flies, bed bugs, body louse, etc. Today we are focusing on ticks and mites. If you come to the homepage of Vectorbase, here in the header, you will find that we have a link to our YouTube channel, and there you can find previous recordings from webinars and workshops. We also have a Reddit channel that we share with the other BRC from Virus and Bacteria, and we regularly post things on our Facebook and Twitter social media. Probably the best way and the one that people most of the time use is this: contact us, and we are always happy to hear from you. Even if this is the first time you hear about Vectorbase and you want to learn how to use it, always happy to hear. Don't believe that, oh, but I don't know where to start, what I'm even going to ask. Say just that and say, what is your project about? I will be more than happy to help you. If you prefer, we also have a help section in which you will find resources, and I'm going to tell you a little bit more about that. To use our site, you don't have to register, but you can here in the top right corner, and this gives you the advantage of being able to save your jobs. So as I said, here in help, you click on learn how to use ViewPath, and you will find these different resources: FAQs, virtual event. You can invite us to your lab meeting, and we will join in a Zoom conference call, and you can tell us what is the projects that you develop in your lab, and we can teach and show you what Vectorbase has to offer for you, or any of the other sites for that matter. Here we have our webinars, and you will find both the recordings of the previous webinars as well as the ones that are upcoming. We also have workshops, and we have the recordings of the previous ones that we recorded, and announcements of the new ones coming up, PDFs with tutorials, which you can also find in the workshops here, and methods. If you are using Vectorbase, you're maybe wondering, how do we process the genomes? How do we analyze the RNA-seq data, the mass spectrometry data? So here you will find the methods. Here on the side of the website, you will find news and tweets. So if you're not much of a social media person and you want to keep up to date of what we do and when we do it, you just open this panel and you can see it. We send you emails normally every 2 months telling you when the website has been updated, but sometimes we also send other emails. For example, we send one for this webinar. But if you don't want to receive the emails, that's fine, and you will also have these announcements here on this side panel. Another way that we use to communicate with you is this banner here, in which, for example, we post the announcement for this webinar. Let's say you decided to create an account for the website, which I already said, you don't need to use the site, but it gives you some advantages. One of the sections of your profile will tell you if you want to receive email alerts from us. I say, oh yes, I want to receive from host or Pyroplasma, Vectorbase, and ViewPath in general. You save and you will receive the emails. If at some point you want to change your preferences, you just come back again to your profile and save again. Okay, now into the data: genomes, transcriptomes, and proteomes. Here in the top of the website, you will find this that says My Organism Preferences, and says 70 of 70. It means that Vectorbase currently has 70 genomes, and you are using all of them for anything that you are doing on the site. You will understand what I mean in a second. If I open this panel, I see that at the top, I have Arachnida, and at the bottom, I will see all the insecta. And here are all the different ticks that we have. We have Dermacentor, Ixodes, Hyaloma, Hemophysalis, Rhipicephalus, and we also have mites. We have Sarcoptes caviae, and we have this cheaper mite, Letotropidium. So you can select 1 or 2 species, or you can actually select them all and always have them available for your queries. Here, for example, it says 12 of 20 because I have selected all these 12 genomes from mites and ticks. So it's telling, out of the 70 genomes that we have available in Vectorbase, anything you do on the site is only working for the 12 you selected, because I have enabled this. So if I come to data, organisms, it's telling me you are looking only at 12 organisms, and here I have Dermacentor and the other tick species. But I can also disable this, and now it's showing me all the 70 species. But you can also filter using the search box here and type, I only want to look at exotic species, and you don't want to do it from here, you can do it from here. And here are the 4 different exotic genomes that we have. So you will notice we have the genus, the species, and the strain to which it belongs. For example, for Ixodes capillaris, we have 3 different strains. The cell line ISE6, we have the new PubLab Hi-Fi strain, and we have Ixodes capillaris white. One column that is very important if is telling you if it's a reference strain or if it's not. Currently, the reference strain for Ixodes capillaris is white gold. But as you may know, this new strain reduced significantly the number of supercontigs. This is a much less fragmented genome. We now have 648 supercontigs. So this will become, in the next release, the reference strain. What this means for you is that we will align all the data, the RNA-seq, the microarrays, the proteomic data, to the one that we call the reference strain. Will white gold disappear? Not yet, but in the future. But if you believe you need still the white gold because you are doing your analysis and you are interested in know when are we deleting this genome from the website, please get in touch with us. But for the time being, we believe it's going to be up at least for 6 more months. Also coming to the data menu, you will find a link to data sets in Vectorbase. So in addition to genomes, we have this specific view here that we just looked at. Here you will find everything, all the different data sets. And those are, for now, for this release, 63, which was in May, we have more than 1,000 data sets. These are the different categories of the data sets. So in addition to microarrays, genome, and with their corresponding annotation, population biology, mass spectrometry data, and RNA-seq, we have other types of data sets, which I invite you to explore. So you can say, okay, from the more than 1,000 data sets in Vectorbase, and you start typing, I want to look for RNA-seq. You click in this arrowhead for category of the data set, and it tells you we have more than 334 data sets. But again, you can enable the filter with organisms and only care about mites and ticks. And so it's telling you we have 807 RNA-seq data sets, and 18 of them belong to RNA-seq. So these are all for ticks, and these ones belong to RNA-seq. But where all this data comes from, we are not a genome sequencing center. We are, as I said at the beginning, we are the home of the data. So the data come from scientists like you. These are direct submissions from scientists generating the data in their labs. And we also do literature curation. So what do we need? You already submitted to INSDC, let's say GenBank and CBI. You don't need to submit to us again with a complicated format. You just send an email, contact us, and give us the accession number. And from one of those INSDC's database, we copy the data. That's simple. That works for the genomes and for the RNA-seq and microarray data. If you are working with mass spectrometry with proteomics data, we just need a database file, could be in Excel, and in one column you put the gene ID, and in the other one the sequence of the peptides. If you have more information, that's great, and that's all we need. And for the population data, I will let Sam tell you what we need, but at the bare minimum, the GPS coordinate of where in the field you collected the data. What do we prefer? That you contact us before a paper publication. And if you already have a draft of your manuscript and you can share confidentially with us the materials and methods and maybe also the results, that will help us process the data. The difference with us and INSDC is that when you submit your RNA-seq data, for example, is up in a couple of hours or from a day to another. With us, it takes at least 2 months, if not 4. So that's why we're telling you, contact us, please, as early as you can. That way we prepare the data. And when you say, okay, the journal has reviewed my manuscript, is ready to be published, at that moment we make it live. We are never going to make live a data set if you are not happy how it shows in Vectorbase, or if you want first your paper to be published. So we will just have in the background, and whenever you said it's ready, so we'll make it ready, but we need time. How do you know which data sets are coming? Come to data, data sets we are working on. And right now we are on release 63. So you will see everything that is 64 and up. And you can check, okay, I'm working right now with this tick, and I found this very interesting experiment in which they were blood fed, and this blood had parasite, and I really want to analyze this. And you come and check, oh, Vectorbase doesn't have it. So you contact us and send us the link to that data set. It doesn't have to be your data. Maybe you found it in a paper and you want for us to load it. That's great as well. Okay, and here, after that introduction, I will switch gears, and now I will leave you with David Bruce. So the only thing that I want to say before handing this over to David is that he is going to talk about a tool called Apollo. David.
All right, thank you very much. Thank you very much, Gloria. Let me just share my screen and tell you a little bit about some of the things that we're going to discuss today. I think you can see my screen, which is a PowerPoint view, not in presenter mode. Is that correct?
Correct.
I thought I would start with this because, as Gloria has indicated, the ViewPathDB family of databases provide very powerful tools for being able to mine and explore data in a variety of ways. And we won't have time to be able to go into all of these resources, but there is extensive material available online through YouTube tutorials and all of the resources we've made available from webinars and face-to-face workshops. And as Gloria indicated, because the architecture is the same, the tools and exercises that have been developed for Plasmodium or Babesia or Anopheles also apply equally well to Ixodes or Theileria or Giardia, whatever organism you may wish to look at. What you're looking at now is a sort of anecdotal, arbitrary top 10 list that I assembled to try to highlight functionality that is perhaps underappreciated on the database. This includes functionality that is pretty widely known, but perhaps could be more widely known, to be able to visualize and interrogate expression profiles, for example. And we'll take a look at that today. To integrate diverse data, leveraging orthology for information across species, analyzing your own data, and most importantly, opportunities to obtain further help. Now, perhaps the most powerful functionality that's available, it allows you to be able to ask your own questions and explore the data. And I'm going to give you an example of that in what we call a query strategy. So if I turn to look at this live, what we can see is a strategy that I had assembled last night, just attempting to try to look at some of the new assembly and new annotation available for Ixodes scapularis. As Gloria already indicated, the reference genome has been the YCal annotation. A new assembly based on long-read sequencing from the PAL lab has greatly improved the quality of annotation that is available for ticks. And I wanted to ask what the similarities and differences were. In the reference annotation, we see 23,000 genes that were annotated. And not surprisingly, those map to 23,000 genes. Essentially, all of those genes are identified in the new annotation. We could explore the 100 or so genes which seem to have disappeared. But when I transform these into their mapping, their orthologs, or more properly, not really orthologs, but new annotations in the new assembly, we can compare that with the annotations that were published in this paper from the new assembly from Ixodes, annotating 38,000 genes, identified 15,000 genes not previously recognized in the genome. We can see what those genes are by modifying the Boolean operator here to subtract those. And I'm now looking at the 15,000 genes that are new in the new annotation. And when I look at those genes down below, I see that the majority of those are pseudogenes or tRNAs or uncharacterized proteins. And just because I would like to try to understand the data set a little bit further, I've added some additional steps, removing text terms for pseudogene or tRNA or ribosomal genes or uncharacterized genes. All of these are certainly genes that warrant curatorial attention, but I'd like to focus my attention on genes which we know are expressed and have some reason to believe are in fact functional. So I have asked for those that have interpro domains and such as PFAM domains, and that leaves us with about 1,000 genes, and turning on one expression experiment, an expression experiment of Ehrlichia infected ticks. I've asked for genes that are ever expressed in the top 20% of genome. Interestingly, I find about 20% of the genome, 9,000 genes that are identified here, suggesting that in fact, most of these genes are in the most abundant genes in all of the samples in this particular experiment. This brings me down to a smaller number of genes that I might want to look at. Now, if we take a look at what those genes are, this query is now rerunning as in the most up-to-date data, although nothing will have changed from last night. And as we wait for the results to load, we'll be able to look at individual genes that I'd like to dive into to be able to explore some of the information on gene pages and on the annotation. So we can see that gone now are all of the tRNA genes and pseudogenes and uncharacterized proteins. And we have a number of proteins that are at least annotated as having names. And scrolling down this list last night, I identified a couple of genes I was interested in and flagged them by putting them in my shopping cart here, which I might want to work on later on. Two row interacting kinase type proteins that appear to be tandemly repeated in the genome. So we can dive in and take a look at this at one of these individual genes here and see this citrin row interacting kinase protein. And I can see from the expression profiling data that this gene is annotated as having numerous exons, and there is strong support for those exons from multiple RNA-seq experiments. What we're looking at here is the RNA-seq data from all available experiments showing 7,000 independent experiments, 7,000 independent short-read sequences, or perhaps some long-read sequences that span this particular intron and this particular intron and this particular annotated intron. And we can see that the consistency from the annotation to the read data is extremely good. We see a few reads that were misidentified on the opposite strand, probably because of library preparation issues, but overall the annotation of this gene looks pretty good. I can see that the gene is expressed in, and particularly expressed in this Ehrlichia infected sample. And in the summary experiment, when I click on it, I can see that this gene is most abundantly expressed in infected ticks, something that I might want to explore further. Recall that there were two genes that appear to be tandemly repeated, and if I zoom out a little bit, I can see the adjacent gene here, also annotated as a citron row interacting kinase gene. But when I look more closely, I see that in addition to the proper annotation of the introns that are identified, I see an additional intron between these two genes that for which there is strong evidence, 3,800 reads, suggesting that in fact, these may not be tandemly duplicated genes, but may be a single gene which should be merged with an intron that hasn't been annotated. I can explore that further by looking at the orthology information. These genes have been mapped to ortholog, this particular ortholog group in the OrthoMCL database. And if I jump over to look at the OrthoMCL database, I see that this gene is represented in essentially all metazoa usually in a single copy gene in the individual species, although there are some exceptions. And when and that they are typically 1,800 amino acids long, for example, in 80 species. I'll filter this down to look at Ixodes, and now I can see the two genes that I'm looking at. In Ixodes persicautus, this gene is annotated as, there's a single gene with both of the domains that we're looking for, but these two tandemly repeated independently annotated genes in Ixodes scapularis look for all the world as if they are actually two halves of the same gene, confirming my interpretation. I can, from the gene page itself, I can leap to the genome browser, providing me with access to hundreds or thousands of tracks of evidence that I can easily turn on or off by clicking on this information, asking, for example, about comparative genomics. I can turn on the syntenic sequences, which will now show up down at the bottom of my screen. And I can see that, again, these two genes in Ixodes scapularis have been annotated as a single gene in another species. So I would like to be able to examine that, and by turning on various different expression profiling data sets in different, as I see that these particular genes seem to be expressed not in uninfected ticks fed or unfed on blood, but shows up positively in ticks that are infected with Ehrlichia or Anaplasma, or I believe we have Borrelia infection. So if I'd like to be able to explore this further, I will go back and annotate this gene in Apollo. Turning over to the Apollo annotation platform, you'll see a similar expression, a similar browser view, allowing me to zoom in and zoom out, look at these particular genes, turn on and off the various different tracks that were available, exactly as we did with JBrowse. But we also have the ability to turn on tracks that can be used for annotation. And I have turned on draggable annotation tracks that I will show you in just a moment. The individual transcripts annotated here, highlighting reading frames in red, green, and blue, and the tracks for which we have evidence for introns as indicated down below. So our prediction, our interpretation is that these two genes should be merged into one. So I am going to take these draggable tracks of annotations and move them into a yellow region up at the top of the page for user created annotation. I'm going to take this gene and this gene, drag them both together into the top, and it will create a single merged gene with a problem that's indicated with the exclamation point. I can also see the intron that I would like to be able to annotate that is present in 3,800 instances. I will take that and drag it up into this individual gene where it will be merged in to that gene. And as that happens now, I can, let's see, pull it up again for reference to be able to look at it. And I'm going to zoom in to make sure that the gene has been properly assembled. I can see now that in adding this intron into the, in adding this new intron into this merged gene, that it has been added correctly at one end, but not at the other end as indicated by the red line. And that's caused a frame shift polymorphism that would prevent proper translation of the gene. So I can correct that by zooming in further down to the nucleotide level. And when and now I can see, let's zoom in once more. And now I can see that, in fact, that annotation didn't properly. So I will take the end of this and drag it to reach a consensus splice site that was represented by the individual version here. And hopefully now when I zoom back out, everything will have popped back into frame as indicated by the colored bars. And indeed it does. I will remove this which I was just using for reference. And examine the annotation on the side here, indicating that this gene is now finished as far as I am concerned, at which point it will be incorporated into the annotation so that the next time you go to take a look at this gene, tomorrow, for example, you will see the revised annotation of that gene. Now, I think in the interest of time, I'd probably better stop here rather than showing other examples. I've recently annotated several genes in Ixodes scapularis. It's pretty straightforward to do. And for those that who might be interested in doing this, there are tutorial materials, and you can click on the Contact Us link for additional help if you need it. There are examples of genes that should be fused, genes that should where alternative splice products should be added. I'll leave you with maybe just one example of a dramatic gene that I took a look at yesterday. Going back to the annotation that I looked at here, this is a gene which is annotated as having 45 introns and 20 alternatively processed transcripts. But when we look at the RNA-seq evidence, we can see that in fact there is extensive evidence for alternative exon skip polymorphisms. These cassette exons that are shown in the sort of repeating blue and red and green R's here represent in frame, I think 29 amino acid exons that can probably be added or removed in a variety of different ways. If there are 40 of these exons that could be added or removed in all possible combinatoric patterns, that would leave 2 to the 40th, or roughly 10 to the 12th different transcripts. And I don't think any of us wants to see 10 to the 12th possible transcripts annotated on a gene, highlighting the importance for researchers, if you're interested in this individual gene, to be able to dive in and look at what the experimental evidence is to see whether any of these transcripts is in fact appropriate. You can see that I have added user-created annotation for this gene representing all of the possible exons, but I am not going to take the time to annotate 10 to the 12th different possible transcript variants, which might be possible based on splice junction acceptance. So with that, I think I will stop and turn the back to Sam and Sarah, who are going to tell you a little bit more about tick data and the availability to view that data. We can, of course, talk more about this in the question time or through your comments and questions online. Thanks very much. Back to you,
Thank you
Gloria.
very much, David. Sam?
Hi. Thanks, David and Gloria. So I'm going to show a little bit about our maps. So I just first want to highlight that this is our current mapping system, but we're about to release soon an even more exciting system with new features and more functionality and the ability to have a lot more data. But everything you can see now, it'll be even better in the new system. So I just want to highlight some things. This is our mapping system. We have our filter box up here where you can put search terms, and you can also facet data using this faceting window down here in the right-hand corner. And so I've just put Ixodia here, and you can see geographically where we have data from, example, the United States. And we can see available data types for this geospatially located data, including pathogen status and abundance. So I'm just going to, for example, switch to pathogen status. And you can see we have data from a number of tick-borne pathogens. And I'm going to, for example, switch to infection status and say I just want to see ones where they did an assay and the data was there was an infection, or the infection, virus or bacteria was present. So I'm going to click here, and you'll notice that that adds the presence filter here. And then I will go back to pathogen, And now let's say I want to do Poisson virus, for example. And now obviously there's a lot more data that exists in the US for this, but uh, a real focus of tick field data is something that we're working on. And for any data set, you can click on the data point and see the collected metadata, including I can even, so this was a Poisson virus detected in Ixodia scapularis using real-time PCR from a nymph of unknown sex. And if we want to know where this data came from, we can click on this project link out button. And we can see that this particular data set was from this paper. Now, most of our tick data is actually currently from large metadata analyses or publications where they have went through and done surveys across the United States. But what I wanted to show you next is if you kind of want to see the direction that we're going, this is mosquito data, where we have many more data streams, usually or often in the United States, directly from mosquito abatement districts. So these are the governmental entities that are going out and controlling mosquitoes on a day-to-day basis. And from some of these mosquito data sets, we have, for example, years worth of their regular surveillance. And so this one goes back to 1980, for example. And with data sets this large, we often have, instead of county level data that we have for a lot of the tick data, oftentimes this data is trap level. And so you can see here we have a multi-year trap level data set. Can't scroll. And here we have monthly. You can even make the resolution even finer. And as we continue to zoom in, you can see we can actually get down to that trap level data. And as I mentioned, that we're about to release some new data sets. And so we're going to start to have some data sets a little like this, like highly focused repeated collection data for ticks. So you'll be able to use some of the tools that I'm showing you right now in mosquitoes and visualizations for tick data. So we're very excited about that. And that will include longitudinal data for pathogen testing. So again, I'm going to show a mosquito example, but this is from the South Las Vegas Health Department, and they do regular surveillance for a variety of pathogens. So again, I'm going to go to pathogen and do West Nile virus because I know that they have more of that data. And when I go back to pathogen status, infection status, we can see some longitudinal quantitative over time measures of the pathogen status. So the bars are they performed a test, and the line here is what the positive tests were. So interestingly, they seem to have a high year of infection here, infection status, but a low number of tests, and they increase the number of tests, but seem to have less positive. So it went from a year of low West Nile to a year of high West Nile. Now, our new system is going to show one screenshot of this. Will allow for really rich metadata. So this is the data set that we're working with right now, and you can see it has significantly more metadata fields than our existing system was able to visualize. So if you have a data set and you also collected, where it was collected or what host it was collected on or what the host was wearing, or really any kind of other metadata, our new system will be able to have that. And again, as I mentioned, our tick-based field collection longitudinal data and geospatial, geotemporal data is something that we're really pushing forward to. So if you have any exciting new data sets that you've just published or are planning on publishing, or perhaps you conduct regular surveillance, we would love to work with you. Just please contact us. Especially, some people do regular tick flagging, for example, or citizens submitting submitting ticks for tick testing. So these are some of the projects that we're beginning to work on curating, and we'd love to reach out and about sharing this kind of data on our site. And obviously too, if you would like help utilizing this data or accessing this data, do reach out to me or Sarah using the Contact Us button, just like Gloria showed you at the start. And I'll pass it back to Gloria. If there's no, let me see if these are any questions.
Thank you, Sam.
Yeah.
Okay, so I'm just going to summarize very briefly so we can hear more from you guys. I'm very happy to see the chat has been had lots of questions. So what David just showed us was a tool called Apollo, and this is important because the better we have the annotation, the better will be the predictions that we do with RNA-seq, with transcriptomes and proteomes that we have on the site. And he showed you how to use Apollo and how these tracks of the genes combined with the evidence that we have is how we are able to refine these gene annotations. He also showed us how to construct different strategies. So if you go to My Strategies and you will click in public, right now have 26 different strategies, and you can click on any of those and start adding with Add Step the different questions that you want to ask for your own experiments or research. And later we hear from Sam, and he was telling us about the population data and tools, MapView, and he was showing us how we host data, worldwide data, different types of data sets, and how we had most of the data for mosquitoes, but we are right now, Sam and Sarah as well, are doing really hard work to not only improve the map, but have more representation of tick data. And now I open the floor to discussion to hear from all of you. I don't know if you want to probably use the reactions and raise your hand, or keep typing in the chat, or just open your camera and start talking.
Should say that we have been all of us have been responding to comments in the chat, and will continue to work through there. There are a number of comments from people about loading data, and certainly the easiest thing to do is to click on the Contact Us link to provide information. We'd encourage you to click on that Contact Us link rather than emailing Gloria or any of us, because that way it enters into a tracking system and we can make sure that it gets to the right person. If your data involves tick collections that you'd like to see represented on the MapView visualizations that Sam showed, or the newer MapView, that might wind up being routed to Sam or Sarah. If you have questions about how to re-annotate a gene, it might go to myself, or more likely to one of the curators who's working on the project. If you have questions about data sets that would be prioritized for loading so that others can take advantage of them, that might go to Gloria or others. So we encourage you to do that, and I will get back to the chat to be able to follow on some of the responses that that Sam and others have been added.
Anybody from the attendees wants to participate or say something? Ask or give us feedback, or
just reading down the list, I see, for example, questions about population data. I see questions, comments about proteomics data. ViewPathDB databases do support proteomics data. I don't believe we have any data currently loaded for Ixodes scapularis.
Yeah, for ticks we have mass spec. Ixodes scapularis we have mass spectrometry data.
Okay, I was not seeing that in the browser views at the moment, at least not for scapularis, but we can take a look at that, and certainly that is something that we can support quite readily. So again, we're happy to talk with you further about the nature of the data you have and how to represent that. You might imagine, for example, that in the browser views that I showed in evaluating a couple of gene models earlier, I might want to turn on tracks indicating proteomics data, to see whether and when these are expressed, or maybe even sometimes whether we can see support for gene models and proteins that span annotated introns, supporting or refuting one or another different annotations of transcripts.
And one of the suggestions that was made in the chat, which I think was really good, was having like a banner or announcement on the side of YCOL saying that, that this reference genome will be retired, and, please work more with the new pseudoscapularis. That's a really good suggestion. Thank you very much.
Yeah, I had also responded to that, as well as we have with other genomes. Well, as Gloria indicated, the old annotation will no longer be considered the reference, but we will take the previous annotations and map them to the new assembly, so that that information is available on a track in the browser, allowing you to compare old annotation with new annotation. That makes it possible to see where things have changed. And well, in the majority of cases, it's clear that the better assembly and newer algorithms has led to better annotation. There will also be some cases where the prior annotation was correct, and it's helpful to have that as something to view. I see that you have turned on your camera, Travis. Did you have a question?
Yeah, thanks very much. So I appreciate this webinar. It's been really informative. We have quite a few tick species that we're doing assemblies from that are in various states of finished versus, almost there. And I'm kind of wondering, at what point do you want to see it? I assume when we're pretty much done with what we're going to do is when we should pass it to you, as far as assembled genomes. Is that correct?
I was going to ask, are you planning also to submit to NCBI for annotation? You're doing the annotation. How are you going about that?
So it depends on the species. So we have a couple of different things that are in play. Some species we have gone ahead and annotated. Others, the assembly is basically done, and we're at the decision point of what's the best thing to do going forward for annotation. And so some of those we may submit to NCBI. One of the questions would be if you all would like to do that, or if that's not something that you do. What's your recommendation?
Well, we prefer when NCBI get it submitted. We had some, our users have had some issues when they annotate the data. Later, they have trouble uploading that annotation to NCBI. So that has delayed for us to load the genome in vector base. So if you already have the assemblies, I suggest you go with the NCBI annotation pipeline. And from there, it's very easy for us just to copy it over.
May I say, though, we'd still love for you to maybe email us and kind of give us the status of each one. And sometimes we can help nudge NCBI to, if it's like, oh, that's more important, they'll listen and try to help you cut the queue sometimes if it's missing.
Early communication is always helpful. It's good to know what's available and what it is that's coming. And just for clarity, as is probably clear to you, Travis, but also to others on the call, we are not an archival repository. We are supported by grants and contracts from various different sources around the world. And while ViewPathDB resources have been continuously supported for 20 years, no one has made us the guarantee that data will be present for the next 200 years. Certainly none of us will be here working on the project 200 years from now. It's therefore important that data be deposited in archival repositories such as GenBank or Ensembl. And that also makes sure that we and everyone else in the world at Ensembl, at Swissprot, or elsewhere is working from the same reference. So our very strong policy is to pick things up from your GenBank deposition, and we'd encourage you to do that for reasons I'm sure you appreciate. That's certainly true for the assembly. Annotations are a little bit trickier, but also very beneficial. It really helps everyone if the proteins that are available in VectorBase are the same as the proteins that are available in SwissProt, or the proteins that are linked to from structural genomics initiatives. And as Sam says, if you encounter problems with QC in the GenBank submission and approval process, that may be something that we can that we can help with.
All right, I have one other completely different question on mites. Is that we have a variety of mite projects that we're collaborating on, and several of them involve doing sequence capture, and some of them are from scrapes of individuals with mange. So it's a community of mites. Do you have the ability to deal with data that's essentially like pool-seq?
Yeah, so I can, so this is an emerging theme, the general issue of metagenomic sampling, whether it's from an environmental sampling or populations or this kind of issue, and it depends a lot on the consistency and clarity of the data. First of all, I should mention that that VectorBase certainly is committed to providing information for on mites and for the arthropod community in general, while we are supported while we are supported primarily out of concern for human public health issues. We recognize that that all of these organisms have things to say about each other, and so we are certainly interested in data on mites and even on non-human feeding ticks, for example. As far as the annotation is concerned, I think it will depend on the data, and that's where discussion may come in. The OrthoMCL database does a pretty good job of handling and properly positioning fragmentary assemblies and partial genes to be able to identify what is there. And so many people in the environmental sampling community have asked us about dealing with metagenomic or even metatranscriptomic data. So far, it's not something we've done, but it's worth discussion.
Good. Thank you very much, Travis, for your questions and your feedback. I see no new questions in the chat. I think they have all been replied to. So
one thing, Gloria, that might be worth mentioning, I noticed, for example, in the most recent or near, not quite most recent, several comments have talked about research projects, thesis projects related to RNA-seq analysis. And one of the bullets on my possibly underutilized list of resources is the ability to analyze your own data using a user-friendly cloud-based interface to or a web-based interface to a cloud-based Galaxy platform, which we have automatically populated with all of the reference genomes, for example, for the Hi-Fi Ixodes sequence. If you have RNA-seq data, you can map that against and using this Galaxy pipeline, and there are tutorial materials that are available for use. I should also mention, as many people have probably seen before, that that these query strategies are can be used to share your analyses. That long choo-choo train of that I'd use to identify genes for possible annotation changes is saved as a query strategy. I'm saving it right now, and in the next couple of minutes, there we are. I will grab this link to paste into the chat, and we can, I think, Gloria, maybe we can send it to people who have who have registered. But if anyone who would like to click on that link, we'll pull up exactly the view that I had highlighted of 99 genes in the new annotation for which we have high confidence based on their domain architecture and expression are functionally significant, and for which there is evidence that from RNA-seq data that the annotation may be slightly incorrect and should be reviewed.
And to that point that David makes about that you can analyze your own data in the cloud. So I remember at the beginning when I was giving you the outline, I said, we really want to repeat what we told you last year in the TAPE webinar. So if you go back to the recordings from last year, we have one webinar about vector-based and another one about pyroplasma. And if I'm not mistaken, the one about pyroplasma, it has the details about how to do the galaxy analysis in the cloud. So I, once again, taking this David comment, go back. If you didn't attend last year, or if you want to refresh your memory, watch the recordings from last year, vector-based and pyroplasma. And with that, we reached the top of the hour. We really appreciate you coming here. And thank you very much to David and to Sam and Sarah also answering the chat questions in the background. And of course, to all the people that attended, all your questions, we really appreciate it. Thank you very much. And hopefully we'll see you next year for the TIC and Minds 2024. Thank you, everybody. Bye-bye.